Hello, BISC132. This is the beginning of recorded lecture 3.1. Still on plants, but not for long. I know plants took up the majority of the previous exams material, but there's just one last topic in this plant form and physiology chapter that I think is really, really important before we can be done with plants. And that is the transport of water and solutes, how stuff moves around. Now, if you read this section in the textbook, there's a lot of math here, but I'm going to try to give you the non-math version of this because I don't think understanding the equations and all that stuff is really important in an introductory class. So a concept that we're going to run into as we go through this is the concept of water potential, which, if you try to break it down into the non-math version, it is very simply the potential energy of water. It's kind of a weird thing to think about, that water just existing has potential energy. The take-home message regarding water potential, what we're going to bring up later, is the idea that water moves from high water potential to lower water potential. So keep this in the back of your mind as we're talking about things moving around and water potential changing. If something has high water potential, that means there's going to be a tendency of water to move away from it to areas that have lower water potential. So just keep this statement in mind. It'll definitely come up later. So there are actually two systems involved in the transport here. There's one system that deals with transporting water, and then there's a separate system that's involved in transporting solutes, dissolved sugars and stuff. Let's talk about water first. So this is going to be a ground-up transportation system. Of course, water being absorbed in the roots from the surrounding soil and then sent upward toward the leaves that need that water to do photosynthesis. So this is always, from down to up. This is going to involve three separate stages here that work in three different ways. So let's start this journey of water. Step one, from the soil to the root cells. So let's zoom in on this part of the figure here. Okay, so the way this works has to do with, as this says, water potential. So by manipulating the concentration of solutes in these root cells, by making sure that there are a lot of just dissolved sugars and stuff like that in these cells, that creates low water potential. Remember what I said, water moves from high water potential to lower water potential. So if you create root cells that have a low water potential, water's just going to move from the surrounding soil into those root cells. It's as simple as that. So increasing the concentration of solutes in the root cells creates low water potential, therefore water moves to the root cells from the soil. Okay, that was simple enough. Now we have to go up. So this is from the roots to the stems. This process, if we zoom in here, this process is going to involve, as this says, cohesion and adhesion. You might remember this from BISC130, cohesion and adhesion. These are both defined in the key terms. Cohesion is the tendency of water to stick to itself. Adhesion is the tendency of water to stick to other things. And these forces together are going to result in what's called capillary action, where just because of the tendency of water to stick to the stuff around it, if you have a tube in water, this water is going to fight against the force of gravity and rise up the tube because it's wanting to stick to the walls of that tube. This is an artificial setup with a glass tube and a beaker, but this is exactly what we see when we look at the tubes of xylem, for example. So yeah, the water is going to crawl up the walls of this xylem because of cohesion and adhesion. Simple as that. So even though these were defined in earlier chapters, they should be in the key terms for this chapter as well. So from the roots up through the stems, cohesion and adhesion draw water up the xylem. And finally, we need to make it from the stems to the leaves, because again, those are the cells that actually need this water. Photosynthesis consumes H2O, and so these are the cells that actually really need it. Zooming in on this, this is the weirdest process, or weird in my opinion. This is going to involve something called transpiration. So, transpiration is where water evaporates out of these leaves. It's lost, it goes away, it exits the plant body. And what that does is that causes negative water potential in these leaves. And again, to go back to this slide, remember what we said, water moves from high water potential to lower water potential. So if these leaves have low potential because they've lost a bunch of water through these stomata due to evaporation, that is going to draw water from the stem, from the xylem, into these leaves. And this kind of sounds stupid. I mean, the whole point is that these cells need water. You're getting water by losing water, and that's exactly what this is. I mean, I guess there's a saying in the business world, you got to spend money to make money, and that kind of rings true here. This tissue acquires water by losing water. So a fun statistic is about 90% of all water that a plant absorbs initially, about 90% of it is just lost due to transpiration. Not used for photosynthesis at all. 90% of it just gone. But as weird as this is, it works. There is a net gain of water. Water is used for photosynthesis. It's just giving it away, having it evaporate is what transports it to these cells. So here's my summary of this from stems to leaves. Transpiration causes negative water potential in the leaves. Therefore, water moves, from high to low water potential from the xylem to the leaves so they can use this water. So weird to wrap your brain around that they're wasting water to get water, but it works. Okay, so this was half of things. This was the xylem. This was the transport of water. Now we have to talk about phloem and the transport of dissolved sugars. Now, before we do that, there are some terms that you should be familiar with. Photosynthesis in these leaves produces what is in broad terms called photosynthates. So when we're talking about sugars like sucrose, they just fall under this broad term of photosynthates. So that's what we're talking about now, the transport of photosynthates. These cells producing these are called sources. So leaves, for example, are a source, and they are transported to areas of the plant called sinks. So these are just areas of the plant that need those photosynthates. This can be roots. They're obviously not doing photosynthesis and they're actively growing. They need energy. Young shoots, they require an input of energy because they're actively growing really fast. Developing seeds or fruits, they definitely need an input of energy. And so this translocation process is what's going to transport and then here's the sentence that pulls in all of these terms together. Photosynthates are transported from sources to sinks. Hopefully that made sense. And the important thing is that this can be up or down. So the transport of water was always up. But if you're going, from leaves to roots, obviously that's down. But if you're going from leaves to developing seeds or to younger parts of the shoot, that can be up. So this process of translocation is not always in a single direction. That being said, in this particular example I'm going to use, it's showing the source up here and the sink down here, but it could be either direction. Okay, so just as with the transport of water, we can break this into three stages. The first stage here is moving from the source cell into the phloem. This process involves what's called active transport. And hey, maybe this sounds familiar to you. We talked about active transport in BISC130. This is a very different example from neuron cells, but active transport spends energy. You're expending ATP to move stuff from one side of a membrane to another. So we're spending energy to do this. I say we, but plants are spending energy to do this, but in the end, it's going to be worth it. So this process called translocation, sugars are moved from the source to the phloem by active transport, and this costs energy. Okay, Now, you may have noticed that, this figure about translocation, it's supposed to be about movement of the phloem, but this figure also has the xylem labeled over here. Well, if you'll remember, xylem and phloem are paired with one another in vascular bundles, whether it's a eudicot or a monocot, they're always right next to one another, xylem and phloem. And here's the reason why. Xylem has a relatively low number of dissolved sugars. It's mostly water. And phloem, especially in these parts where we've been actively transporting a bunch of it over, has a high concentration of dissolved stuff. Maybe this sounds familiar to you, discussing the concept of low concentration of solutes and high concentration of solutes. Well, this is where osmosis comes into play. Again, flashback to BISC130. So what is going to occur now in this sort of second step, osmosis, which I've put in the key terms again. This is water moving from low to high solute concentration. Osmosis is going to cause water to move from the nearby xylem into the phloem. And again, this is because they are next to one another. And this doesn't cost any energy at all, but water is going to move from the xylem to the phloem. Now we bring in water potential again. Yes, it's involved in this too. So having a bunch of water come into the phloem is going to increase the water potential of the phloem. Keep that in the back of your head. The phloem is now has high water potential. We're about to be done here. Final step, we got to get these solutes into the sink cells. In this example, it's a root, but somewhere into the sink. And this is going to involve water potential. These sink cells simply have lower water potential than the phloem. Remember, water always moves from high water potential to low water potential, so it's as simple as this. The phloem contents, these photosynthates, move to the sinks because they have lower water potential, and this doesn't cost any energy at all. So hopefully this made sense. I think these figures do a good job of showing this, and I broke down each of these transportation steps into 3 easy stages. But yeah, I think that this is important to understand as a capstone to talking about plants. Now, there are a couple of plant chapters left if you're following diligently along in this textbook, but we're skipping these. Chapter 31 about soil and plant nutrition, and chapter 32 plant reproduction. I'm sure if you take a plant biology class, you'll definitely touch on these topics, but for an intro biology class, just in the interest of time, we have so many other things to get through. I'm skipping these chapters completely in the interest of time. So we are now done with plants and ready to move on to animals. So just like we've done with the previous chapters, before we talk about specific groups of animals, we have to have just some generic background about animals in general. There's going to be a lot of background, though. In fact, this chapter, this short chapter, Introduction to Animal Diversity, is all just sort of background. And so it might seem kind of dry at first, but a lot of this is sort of laying the groundwork, defining these concepts and these terms that we are going to see play out in the next couple of chapters about animals. So, okay, features of the animal kingdom. What is an animal? Well, animals are all heterotrophs. This should sound familiar to you. Here's a heterotroph. A heterotroph, defined in the key terms, an organism that consumes organic substances or other organisms for food, for carbon. There are, of course, several different types of heterotrophs that we see among animals. We see carnivores eating meat. We see herbivores eating herbs, eating grass. Omnivores eating either. A bear will chow down on some tubers and berries as well. And then parasites, that's also a form of heterotrophy. You're stealing energy over a long term, but you're getting it from an organic source. So heterotrophs can be carnivores, herbivores, omnivores, or parasites. Most animals have complex tissue structures. We'll see this play out when we look at the tissue structures of animals in the next chapter. Importantly, no cell walls, extracellular matrix. This might look familiar from BISC130. fungi had chitin cell walls and plants had cellulose cell walls. Animals don't have any cell walls at all. We've got this extracellular matrix instead. It's a looser connection of proteins and carbohydrates instead of a thick, rigid wall. Animals have a diplontic life cycle. So this is not a haplodiplontic life cycle, it's a diplontic life cycle. What this means is only the only multicellular form is the diploid form. So here's a familiar, should be familiar human life cycle showing the diplontic nature here. We've got diploid cells, we've got haploid cells, but unlike plants, the only form that is multicellular is the diploid form. When we go haploid, it's only for egg or sperm. There are no multicellular haploid structures in an animal in a diplontic life cycle. Most animals are motile, meaning they can get from one place to another, but some of them are sessile or sessile, meaning they stick around and they filter feed, or they don't actively move from one place to another. So reusing this bear again, walking around grabbing fish, and then here are some mussels, an example of a sessile animal. And again, we'll use these terms again in the next couple of chapters. Another distinguishing feature of animals as compared to fungi and plants is animals have complex development and a fixed body plan. So what I mean by this is the genetic program for the development of a black bear, for example, going back to this again, is very, very specific from the get-go, from a single cell all the way up to the adult form. There are very complex instructions saying, okay, develop a face that's this size and 2 ears and 4 limbs and all of this stuff, very specific. So this black bear is always going to end up having 4 paws and 2 eyes and 2 ears. It's all very specific. Compared to your typical plant, your typical tree, how tall does this tree grow? It depends. How many branches does the tree have? How long are those branches? In what direction and angle do those branches point? Well, that sort of stuff depends on the environment, the trees around it, where the sunlight is coming in, and things like that. So, and fungi are similar. The body plan that these organisms have is greatly dependent on the environment, but animals, again, in contrast, have a very complex way of always coming out the same way, instead of, it's got a random number of branches from individual to individual because they're in different environments. So that's what I mean by complex development, fixed body plan. Another thing that we see in animals is sexual reproduction. This is, egg plus sperm equals zygote, breaking it down to, the simplest way possible. But we also have asexual reproduction in some animals. So there's our sexual life cycle. And here's an example of asexual reproduction. Again, not all animals can do this, but some can. Here's a sea star having a fragment torn off, growing into a new individual. So animals all have sexual reproduction. Some are also capable of asexual reproduction. Now, I used this figure in a couple of slides ago, but let's look at this more in more depth now. Animal embryonic development. Just briefly looking at this, trust me, there's a lot of detail I'm skimming over, but the first few stages here are, I think, important to understand. So when animals develop, they start with a zygote. It's the result of egg plus sperm. There are going to be a few cell divisions, but importantly, these are cell divisions without growing in size. So this 8-cell stage is about the same size as the zygote. So it's dividing, but it's not growing. So this is called cleavage. From there, the cells are going to grow in size and do more divisions, going further on to the 16 to 32 cell stage. This is called a blastula. The blastula can be hollow or filled with yolk, depending on the animal you're talking about. But eventually, something is going to happen to this blastula called gastrulation. This is where a pore called the blastopore, it's basically an indentation, forms in this blastula. This is, if you can imagine it, because this is still microscopic, this indentation is eventually going to lead to the formation of a digestive tract, and it's going to result in cell layers. We'll talk about both of those things further in the chapter, but for now, here's my summary of this. Animal embryonic development. The zygote divides, like I said, without growing in size several times. This is called cleavage. 1 cell, 2 cell, 4 cell, 8 cell. It's simple enough. The blastula is this 16 to 32 cell stage. It can be hollow or filled with yolk. And then we have that indentation. Gastrulation occurs as an indentation in the blastula called the blastopore. Don't get these mixed up. The blastula is that group of cells. The blastopore is the indentation. Makes sense. It's a kind of like a pore, an indentation, whatever. As that forms, this creates a digestive tract, and this results in cell layers. So again, we'll bring this up again, but I felt like I should introduce these terms now. Okay. And another, I'm sighing because this is complicated. Another thing that I really want to bring up now when we're talking about animals in general is Hox genes. So, as I said, animal development is super complicated and very specific. There are lots and lots of genes that are involved in, telling us how to grow into a very specific body plan. And it takes a lot of genes to, say, to build an arm, for example. It would be incredibly inefficient to have a huge set of genes that, say, okay, make an arm, and then another set of genes to say, okay, make a different arm that's a mirror image, and then another set of genes that say, oh, build a leg, and then another leg. It's much easier to use the same sets of genes for all 4 limbs and just change them slightly. This is, and I think there are probably some computer science majors, in the course, or at least people with a passing familiarity with computer science. This is not unlike a scripting language in programming, calling on, other scripts, for example, to do these things. It's an efficient way of organizing your genes, so you don't have to have a separate set of genes for every little piece of the body. You can broadly use these sets of genes called homeobox genes or Hox genes, applied in different contexts to do different things. And this is true in a human here, this is true in a mouse. And so Hox genes, because animals, especially vertebrates, have so much in common with one another, these homeobox genes end up being incredibly conserved. Our arm is very different from the arm of a mouse, but the genes that we use to build a basic arm structure, there's a lot that we have in common with a mouse. So, okay, how do I summarize this? Homeobox genes, also known as Hox genes, are found in virtually all animals. These are described as master control genes that can turn on or off many genes that are involved overall in the development of a body plan. And here is an interesting way to look at the similarity here when we look at comparative embryology. So these are some embryos of 6 very different animals, but at this stage, they kind of all look the same. It's because they're calling on the same homeobox genes to direct this development. It's only as they go further along in their development that things begin to look a little bit different. Okay, we're seeing a slightly different body plan in these 2 compared to these 4. Let's proceed further with body development. And okay, now they're looking a lot more fish-like over here and looking more like something that walks around on 4 limbs over here. If you want to know what these are going to eventually end up being: fish, salamander, tortoise, chicken, rabbit, human. But again, to go back to this, at a certain point, they all look very, very similar to one another. That's because of these conserved Hox genes in all these different animals, giving them the same instructions. So, again, talking about these homeobox genes, embryonic development in diverse groups of animals, from fish to humans, is very similar because of these conserved genes. Now, And another interesting thing about these is if you start to mess with these, that can really mess with the overall body plan. Because again, these are sets of genes that say, okay, build a limb like this and build it here, and build another limb like this and build it over here. If you start making mutations or alterations or duplications in these things, that's going to profoundly affect the body plan. What do I mean by that? Well, a lot of experiments have been done in fruit flies trying to understand how these work, and this looks like mad science, but really it was just trying to understand how Hox genes work. Here is a fruit fly with legs instead of antennae. So again, just by messing with Hox genes. And here's another fruit fly, normal pair of wings. Here, just a duplication. Two sets of wings, just doubled up on this part of the body. So again, this not just mad science, even though it looks like it. These experiments were done to understand the role of these in overall development. So my point here is mutations in these Hox genes or in the expression of them can result in dramatic developmental alterations, because that's what they do. They're involved in directing development of the body plan. Messing with them is obviously going to mess with the body plan. Okay, so now we're moving on to a section with a bunch of key terms, features used to classify animals. And again, this is going to seem kind of dry, but we're going to come back to these ideas as we go through the various and interesting types of invertebrates and vertebrate animals. So let's talk about symmetry. There are three basic types of symmetry that we see in animals. They are bilateral symmetry. So this is defined in the key terms as a type of symmetry in which there's only one plane of symmetry. So the left and right halves of the animal are mirror images. We've got a beetle here, but you can look at your own body to see this. Humans have bilateral symmetry. Another type of symmetry is called radial symmetry, defined in the key terms, a type of symmetry with multiple planes of symmetry, with body parts or rays arranged around a central disc. So there are many ways that you can draw sort of a plane through this animal and have mirror halves on either side. And yep, here's a coral polyp with that kind of symmetry. And finally, no symmetry at all. So sponges are going to be the exception to a lot of the rules that we're laying down. They're exceptions to these rules, for example, and we'll see that when we talk about sponges. But sponges don't have any symmetry at all. They're more like a tree in how they grow. They don't have this complex development that we've been talking about. So three main types of symmetry in animals. Again, these two are in the key terms: bilateral and radial, and asymmetrical or no symmetry is another possibility here. Now, to go back to this again, one of the things that I mentioned when we have this gastrulation is the formation of cell layers. So here's a way to look at this. This is an animal. This is a cross-section of a tube. So this is really weird to think about, but most animals, with the exceptions being the simple ones like sponges and jellyfish, or sponges, I suppose, most animals have a tube-shaped body. Humans have a tube-shaped body. We've got a mouth, we've got an anus, it goes all the way through. We're basically just a tube. So that creates layers of cells on the outside and on the inside. So one way to have a tube-shaped body is to be what's called diploblastic. Diploblastic, di means two. Diploblastic animals have two cell layers, an ectoderm on the outside and an endoderm on the inside, and this non-living layer in between the two of them. So diploblastic animals have two cell layers. Endoderm is the interior layer. Ectoderm, I'm going to say exterior instead of outside, just to try to not get these mixed up. Anterior, endoderm, exterior, ectoderm. They just kind of sound like they fit with one another. So, use that to not get them mixed up. And it is possible to be more complicated than this, though. So diploblastic animals are sort of the simplest way to have these cell layers. You can also have triploblastic animals, where you have the ectoderm on the outside, the endoderm on the inside, but instead of a non-living layer of stuff in between, you have a third layer of tissue in between called the mesoderm in the middle. So these are tri means three. Triploblastic animals have three cell layers: endoderm, mesoderm in the middle, again, meso, middle, it's easy one-to-one, and then ectoderm on the outside. Again, we're going to see these cell layers come into play in later chapters. Now, among triploblasts, there are three additional groups based on a body cavity. So here's a good figure to illustrate this. And again, all of these are triploblasts: ectoderm, mesoderm, endoderm. The difference between these three worms, and it's not just going to be worms, but the three examples here are all different worms, has to do with an additional cavity here. So it is possible to have no additional cavity. You have the cavity in the middle, the digestive cavity. This is a tube, but if you just have the ectoderm, mesoderm, and endoderm, this is called an acoelomate body. It's possible to have a body cavity here within the mesoderm. So look at this labeled coelom. It's spelled coelom. It is pronounced coelom. It's weird. It is fully within the mesoderm layer. This is important here. So animals that have this coelom cavity are, in addition to the three cell layers, of course, are called coelomates or eucoelomates. And the third way to do this is to have a body cavity in between the mesoderm and the endoderm. So look closely at this. This cavity here in sort of light brown is not within the mesoderm. It's touching the endoderm on one side, touching the mesoderm on the other. This type of body cavity where you're in between these two cell layers is called a pseudocoelom, and animals with this are called pseudocoelomates. So all three of these are defined in the key terms here, and again, these only apply to triploblasts. Acoelomates, eucoelomates, also known as coelomates, and pseudocoelomates. And so obviously the key terms provide a wordy definition of these, but I think a picture is worth 1,000 words in this case, and these are a good way to get these straight about what these three things mean. Additionally, there is a way to break down animals based on this gastrulation event, and this is not going to sound like a very important distinction, but it's going to end up being a major cladistic split, splitting complex animals into two clades based on what this blastopore forms. So, so in a group called protostomes, this initial indentation, this blastopore during gastrulation, the initial indentation becomes the mouth and eventually is going to extend and form the anus. So remember, these are tube-shaped animals. So protostomes, mouth first, then the anus. Deuterostomes are the exact opposite, and there are some other differences in their 8-cell stage, but I'm going to focus on this. Deuterostomes, this initial indentation in the blastopore becomes the anus and eventually extends to form the mouth. So protostomes are mouth first, deuterostomes are anus first. Again, seems like such a small distinction, but it's going to result in two clades because this way of doing things is going to be passed on to descendants, and it's a key way to identify two major clades, two major groups within animals. So here's my summary of that. Among animals with bilateral symmetry and a triploblastic body plan. So this distinction of protostome or deuterostome, this only applies to bilaterally symmetrical animals and to animals with 3 cell layers. Anything else is sort of NA if you're trying to say whether it's protostome or deuterostome. There are two groups based on what develops from the blastopore, protostome or deuterostome, and these are in the key terms, but to put it simply, mouth first, then anus, anus first, and then mouth. Okay, so there's supposed to be a section in here about animal phylogeny, but that's what we're going to do in the next couple of chapters, talk about phylogenetic trees and all that stuff, so I'm kind of sort of skipping this for now. Okay, what we do have finally in this sort of short intro chapter to animals is history of the world, history of animals. So I brought this up briefly when we were talking about protists, but I'm going to introduce this idea again, that animals likely evolved from this group of colonial protists, these choanoflagellates. And here's a typical sponge cell, a choanocyte, and yeah, it looks just like the protist cell. So again, I brought this idea up again, but I'm reintroducing it here. Animals likely evolved from colonial protists, having developed a genetic program for multicellularity. So these protists were not multicellular. They just got together in a colony when there are enough of them around. Animals are all multicellular. They just have a genetic program to make them have to form a multicellular structure instead of just a colony. So there's genetic evidence for this. You can sequence choanoflagellates, you can sequence sponges, and their sequences look more similar to one another than to anything else. And then, of course, the physical traits that I just showed. Sponges share physical traits, especially in these choanoflagellate cells, choanocyte cells looking like choanoflagellates. Sponges share physical traits with these protists as well. So this is the origin of animals from these colonial protists. Okay. Now we can move on to history of the world, and this is way more complicated than it looks, but even so, we don't actually have time to get into this in this particular recorded lecture. This is where I typically run out of time in in-person lecture, so we will talk about history of the world, the evolutionary timeline focused on animals in the next recorded lecture. This is the end of 3.1.